Right. So, good evening, everyone. Can you guys hear me? No. Can you hear me? Right. Okay. Okay then. Right. So, let's start the lesson for today then. So today we'll be finishing off the lesson and most of the important sections or most of the questions or most of the areas that have been questioned in the A-level exam are discussed today, right? So we have to discuss nanotechnology, stem cell therapy, and human genome project, right? So they are not very big topics actually. They are not very big topics, and I don't have anything much to explain actually, right? Actually, godak na explain karana. But there are some things that you need to remember and some small stuff that you need to understand as well, right? Okay, let's start it then. Right. Nanotechnology. Okay, so nanotechnology, from the word itself, you get the idea what it is, right? Nanotechnology is basically a technology where very small particles are used. Right? It's a technology where very small particles are used. Right? So you might have heard the nanotechnology, but the best thing you might have heard, or the most common thing that you might have heard is these carbon nanofibers. Can they have the carbon nanofibers? Can they? Carbon nanofibers are there. Oh, carbon fibers can they? Right. So carbon nanofibers are basically they are these small nanofibers or small fibers designed using this nanotechnology. So ඒක තමයි හිතන ඔයගොල්ලෝ අහලා ඇති common ම දේ. But today we'll be learning about some more applications of this nanotechnology. Okay. So when we consider nanotechnology, first we need to look what nano is, right? What is nano? Nano කියන්නේ කීයද? 10 to the power minus what of meters? Nano කියන්නේ මොකද්ද? Meters හරිනේ. 10 to the power minus what? 10 to the power minus 9. Good, good, good. So it's 10 to the power minus 9. Nanotech 19. Maybe 9, sorry. 10 to the power minus 9. Nanotechnology. Okay, you got it in physics. Ne? Right. So basically, nanotechnology refers to using these very small particles in the level of nano in the development of various medicines, technologies, foods, and all that. Right. So you can see it from the definition. Nanotechnology is an emerging science involved in, this is the definition, nanotechnology is an emerging science involved in designing, building, and manipulating minute structures at the nanometer level, right? So nanometer level like that, 10 to the power minus 9. Usually, we nanotechnology define karanne between 1 nanometer and 100 nanometers. If a certain structure is between 1 nanometer and 100 nanometers, we use those kind of structures in nanotechnology. Okay, so a nanometer is 1 billionth of a meter. Right. Nanotechnology. So, මෙතන තව definition එකක් තියෙනවා, but I don't think it's the best definition. I think the first one would be the best definition. So, there's another definition. Nanotechnology is the creation and use of materials and devices on the same scale as molecules and intracellular structures, typically less than 100 nanometers in size. Right. මට කියන්න, what is the diameter of a eukaryotic cell? What is the diameter of a eukaryotic cell? The diameter of a eukaryotic cell? Here. What is the diameter of a eukaryotic cell? What about the others? I'm getting some answers. What's the diameter of a eukaryotic cell? Right, it's basically 10 to 100 micrometers. But here we talk about nanometers. Micro කියන්නේ මොකද්ද? 10 to the power minus 6. So basically, here what we talk about is smaller particles which are even smaller than cells, right? So they are almost like molecules or atoms or these very small particles which are not even close to the size of a cell, right? So these small particles, these small particles are like molecules and atoms and all that. So basically, when you talk about the hierarchy, or hierarchy in unit 1, molecules make up organelles, organelles make up cells, cells make up tissues, tissues make up organs. So again, nanoparticles, I mean nanotechnology using nanoparticles are at the molecule levels. So basically, you can go from molecular level to above, right? You can rearrange different things. You can redesign different structures. You can uh, initiate or innovate different structures from the molecular level and develop it. Right. So nanotechnology can you get into the molecular level and start developing a certain structure or a design. Okay. So the physical and chemical properties of tiny molecules are significantly different than bigger particles as they have a very high surface area to volume ratio. As the size decreases, the surface area to volume ratio will definitely increase. Right? Take a done, right? Right. American physicist Richard Feynman in 1959 enlightened the world of nanotechnology. Just try to remember this. You don't need to remember it much, but living organisms are built on cells that are typically 10 micrometers across. So it's basically around 10 micrometers to 100 micrometers the size. However, the subcellular organelles are much smaller. Proteins are even smaller with a typical size of 5 nanometers. So, monkey, these nanometers are the size of molecules, right? It's the size of protein molecules, at the size of monosaccharides, it's at the size of lipid molecules. So again, we go down into the molecular level and start constructing something from there. Okay, so proteins are even small with a typical size of 5 nanometers, which is much compared to the dimensions of the smallest man-made nanoparticles, right? So overlooking question about to the structure, they say state the size of the smallest man-made nanoparticle. What would it be? 5 nanometers, right? 5 nanometers is the size of the smallest man-made nanoparticle. Right, so I hope that was clear. So මේක කියවන ගමන් කරන්න මේක මොකද මෙතන මේ ටික ගැන එච්චර ලොකුවට දෙයක් නැහැ. Right. This simple size comparison gives an idea of using nanoparticles as well as very small probes that will allow us to investigate the cellular machinery without introducing too much interference. So basically, when we consider only these microscopes and all that, they actually are limited, right? They limit our scope, or they limit the depth that we can go to. But this nanotechnology goes into the level of molecules, right? Using these technologies, you can go into the level of the molecules and observe from there and get an idea about the cellular levels as well, right? So understanding of biological process on the nanoscale level is a strong driving force behind the development of nanotechnology. Nanotechnology has now become a big business with applications in material manufacturing, energy, electronics, engineering. Basically, electronics using nanotechnology, many small miniature circuits have been built, right? Many small miniature circuits have been built. Okay, so there are so many uses of this. But applications of nanoparticles in biology and medicine are of particular interest. One of the most important applications in nanotechnology is in medicine. Applications of nanotechnology for improving human health is termed as nanomedicine. So our main topic is basically not nanotechnology, it's nanomedicine. Right? I mean, nanomedicine. So the definition of nanomedicine is this: applications of nanotechnology for improving human health are termed as nanomedicine. Nanotechnology can be applied for prevention of diagnosis and treatment of disease. So you can prevent diseases or you can diagnose them, and you can treat diseases, right? So our main topic here is nanomedicine. Mainly nanomedicine, right? The applications of nanotechnology in medicine. That's what we learn here, or in biology, right? Right. Okay. So was it clear what nanotechnology or nanoparticles are? Was it clear what they are? Any idea? Basically, using very small particles uh, in designing, building, in medicine. වගේ දේවල් තමයි nanotechnology වල තියෙන්නේ. So was it clear? What about the others? Right, okay. Then මෙතනින් පල්ලෙහා තියෙන අතන the applications, right? The applications of nanotechnology in basically medicine තමයි. Right? So these are important. Titanium dioxide and silver nanoparticles are used for sterilization of operation theaters and surgical instruments in hospitals. So these nanoparticles are involved in killing microbes, right? So basically they are programmed so that microbes can be killed, right? Right. So what are the two types of nanoparticles? Titanium dioxide and silver nanoparticles are used for the sterilization of operation theaters. So basically, uh, you can either like spray them on the surgical instruments or basically these titanium dioxide and silver nanoparticles are just available in the air in these operation theaters, right? They are just available in the air in these operation theaters where you can sterilize these operation theaters from microbes and all that, right? Sterilization can sterilization can you basically kill microorganisms which cause diseases. Okay, so these two types are important: titanium dioxide and silver. These nanoparticles are able to destroy microbes, right? That's what you call sterilization. Silver nanolotions, right? So, mami number karan the applications. The silver nanolotions are applied in theaters to prevent the entry of microbes, right? So, again, you get silver nanolotions as well, right? Silver nanolotions also they are applied in theaters to prevent the entry of microbes, right? So, silver nanoparticle it's very important to destroy microbes and it's very important for sterilization. Nanoparticles are used in the production of antimicrobial coatings and nanofilters. Right? Antimicrobial coatings and nanofilters. Titanium dioxide and silver nanofilters prevent the entrance of tiny particles such as viruses. So, nanofilters can nanofilters can basically they filter out the microbes. Right? They do not allow microbes to get in. They filter off the microbes and only allow the stuff that is needed to come in, right? Or microbes, viruses, or pathogens, or right? So, what are the two types of nanofilters? Titanium dioxide and silver nanofilters, right? So, make a third deck right? So these nanofilters currently they filter away the microbes. So just know that this titanium dioxide and silver are very important for sterilization. So when we consider sterilization, they have different functions, which include the sterilization of operation theaters, sterilization of surgical instruments. They can destroy the microbes, and silver nanolotions are applied on theaters, and you get titanium dioxide and silver nanofilters as well that prevent the entrance of tiny particles such as viruses. Okay. These nanofilters are used to examine SARS patients, right? SARS SARS full form. But SARS is what kind of a disease? SARS infection. SARS, what kind of disease is SARS? Yeah, it's a virus disease, right? So basically, these nanofilters are used to examine SARS patients. Basically, the doctors or the nurses who are examining SARS patients wear these nanofilters, right? They wear the nanofilters so that the virus from the SARS patient, then SARS, it's something like COVID, right? It's something like COVID, easily transmissible, right? ඉක්මනට පැතිරෙනවා. So basically, SARS patients are examined by these doctors and nurses who wear nanofilters, right? So by that method, these viruses and all that can be filtered off. Then, nano device sensors are used to monitor blood pressure, blood oxygen levels, and hormone concentration. So basically, make a both function එක කියලා නැත්නම් it's also related to nanofilters නේ. So ඒක third එකට යට තමයි එන්නේ. And this is the fifth one, right? This is the fifth one. So, anyway, fourth one, sorry. Nano device sensors, right? Nano device sensors. ඒ කියන්නේ මොකද්ද? These small miniature sensors are put into our body, right? Then අපි ගත්තොත් usually in old people and all that, or very diabetic people, the blood sugar level and all that should be measured almost always, right? The blood sugar level should be always measured. So basically, you can use these nano device sensors which can be inserted into our body. And what happens is, once they're inserted into our body, they can directly give the readings for the blood pressure, blood sugar levels, blood oxygen levels, and hormone concentration. So they are inserted into our body, and they are, since they are very small, they do not cause any issues to our body as well, right? So again, nano device sensors can be used to daily check on our blood pressures, blood oxygen levels, and hormone concentrations. Then nanoparticles can unclog blocked arteries and detect and eliminate cancer cells. Right, this is very important. Unclog blocked arteries, the fifth one, eliminate cancer cells. Right. So basically, nanoparticles got the now. If a blood clot is formed in an artery, these nanoparticles can go and destroy this blood clot, right? The nanoparticles can go and destroy this blood clot and unclog the vessels, right? So it's a blood clot or by cholesterol or something, this blood vessel is blocked, right? So these nanoparticles can be sent to that area where they will start destroying the blood clot or breaking the blood clot or breaking the cholesterols and unclog the blood vessels. And another important function is they can eliminate cancer cells. So these nanoparticles can also, they are very small. These are very small. So basically, inject nanoparticle, we don't get any effect. But these nanoparticles can go, we can like program them to go and search for a certain cancer cell and destroy that cell only. Now, අපි ගත්තොත් chemotherapy. No, for cancers, usually what is recommended is chemotherapy. But chemotherapy has many side effects, right? අර කොණ්ඩේ එහෙම යන්නේ. නැතෝ මේ කැන්සර් හැදුනට පස්සේ තට්ටෙන්නේ නැහැ. right? කැන්සර් හැදුනට පස්සේ තට්ටෙන්නේ නැහැ. කැන්සර් හැදුනා කියලා තට්ටෙන්නෙත් නැහැ. right? ඔය people become bald because of chemotherapy. right? Chemotherapy. Chemotherapy කියන්නේ මොකද්ද? What's chemotherapy? What's chemotherapy? What's chemotherapy? Yeah? Chemotherapy, right? Chemo receptors කියන්නේ මොකක්ද දන්නවනේ? අන්න ඒ වගේ තමයි. Yeah. Chemicals are injected into our body, right? Chemicals. Chemo කියන්නේ chemicals කියන එක, right? So, chemotherapy වලින් side effects ගොඩක් එනවනේ. Your hair is lost, your immunity system is very highly affected. ඔයගේ side effects එනවා. But what these nanoparticles do is, with very low side effects, they can directly go and search the cancer cells only. දැන් chemotherapy වලින් sometimes they might affect your own cells. ඒකයි ඔය කොණ්ඩේ එහෙම යන්නේ. Right? But these nanoparticles can directly go in search of the cancer cells only and destroy those cells only. Right? So that is a very important practical application of medicine where nanoparticles are used. Right? How to check the blood pressure after inserting nanoparticles? Maybe in the nanoparticles, basically are pressure detection පොඩි sense එකක් දාලා වගේ. මම කියන්නේ එක nanoparticle එකකින් කරන්න බෑ, but basically you can create a very small miniature device which detects the change in pressure, and by that දැන් හිතන්න නිකන් aorta එකට වගේ පොඩි insert කරනවා පොඩි pressure sensing device එකක්, and by that you can detect the blood pressure. අන්න ඒ වගේ දෙයක්. That's how they detect the blood pressure levels and blood glucose levels ගත්තොත් the they might produce a miniature device using nanoparticles which can detect these chemical changes in the blood, right? According to the glucose concentration in the blood, these miniature devices might get stimulated. So again, these devices can be produced. Do they use that cancer nanotech in Sri Lanka? I don't know. Actually, I don't think so. I don't think so that they use nanotechnology very well in Sri Lanka. Okay. Right. Several nanotechnology-enabled drugs are available in the market, primarily in areas of cancer treatment. Scientists have developed smart drugs using tiny nanoparticles such as gold particles. Using tiny nanoparticles such as gold particles that can be introduced into the body to seek out and target special cells such as cancer cells to deliver a cargo that would destroy those that are damaged rapidly, right? And effectively in a silent manner with very few side effects, right? So basically, this is how nanotechnology is used to eliminate cancers, right? You can program these nanoparticles and give them a certain cargo. Cargo එකක් දෙනවා කියන්නේ මොකද්ද? What's a cargo? Ships have cargos කියන්නේ like cargo කියන්නේ a certain chemical is given into them, right? These nanoparticles are නිකන් හිතන්න it's like a these gold particles are taken, right? Gold particles are taken and a spear is made out of them and ඕක ඇතුළට දාන පොඩි chemical එකක් that would kill the cancer cells, right? So they are programmed to go in search of the cancer cells and attack them only. And then only if they found the cancer cells will cancer cells හම්බුනොත් විතරයි they will release the cargo and destroy the cancer cells, or else they will not, right? So again, these tiny nanoparticles can be used to eliminate cancer cells. Was that clear? ඒක තේරුණාද? Is it clear? These applications මම මේ කියපු ටික තේරුණාද? Or do you have any doubts? Is nanotechnology a possible essay question? Yes, actually. මොකද මේක ආවා nanotechnology වලින් ගොඩක් questions ආවා. I remember in the 2020 A-level structured essay paper. Nanotechnology වලින් questions ආවා. 2020 A-level structured essay paper එකේ. So yeah, I'll have to check with the guessings. මට දැම්ම කියන්න අමාරු ඉතින් possible essay එකක්ද කියලා. But towards the end, I can confirm whether it's a guessing or not. Right. So was it clear for the others as well? The scientist and the year is not very important actually. I don't think they ask that. මේක නම් ගොඩාක් important නේ. පාඩම් ගැන වෙන මුකුත් නැත්තම් ඔය පාඩම් කරගන්න. But I don't think they'll ask that. Even in the plasma membrane year එකේ වැඩි අහන්නේ නැහැ නේ. year එකයි කවුද හොයාගත්තේ කියන එක අහන්නේ නැහැ නේ. Right? So ඒක නිසා not very important. Right. So spherical nanoparticles consisting of dielectric. What's dielectric? What's a dielectric core? What's a dielectric core? Physics චුට්ටක්. What's a dielectric core? Yeah, what's a dielectric core? What's a dielectric core? දන්නේ නැද්ද? Dielectric core එක කියන්නේ මොකක්ද කියලා? Dielectric කියන්නේ basically insulator එකක් කියන එක, right? Dielectric means insulator. So dielectric means insulator. So dielectric core එකක් කියන්නේ insulating core එකක් කියන එක, right? So spherical nanoparticles consisting of a dielectric core called nanoshells are also used in the treatment of cancers. මාත් ඇත්තටම ඒක නම් කොහොමද වැඩ ගන්නේ කියලා දන්නේ නැහැ. Let me show you a nanoshell. So you can see the nano shell, right? So basically, the dielectric or sulfur, silicon dioxide wage insulating particle, insulator particle, you can cover it with gold and form a small nano shell, right? This entire structure is called the nano shell. So basically, the dielectric core like silver dioxide is covered by something like gold to form a nanoshell that can also treat cancers, right? A nanoshell is slightly bigger than a poliovirus. Gold nanoshells are used in bioimaging enhancements as well. So bioimaging enhancements can basically are to enhance the microscopic images again, to enhance the microscopic images taken by these electron microscopes and all that, something like that, right? Then in the treatment of diabetes, a nano device is attached to the body arm. Monkey are sensors right? Small sensors which can act where the nano devices are used as small sensors. And there are nano devices which is attached to the body which can release the required dosage of insulin required time intervals, right? Sixth one, this is the seventh one, right? Nanoshells. Nanoshells are the seventh one, and bioimaging enhancements are the eighth one. Then in the treatment of diabetes, a nano device. So you can create a nano device by these nanoparticles, which is very microscopic, and you can insert it into your body, and that can be used to release the required dosage of insulin. Uh, in a patient who is diabetic can basically insulin හරියට function වෙන්නේ නැහැ කියන එකනේ. මේක හැබැයි මෙහෙමයි. මේක not to both diabetic patients. Diabetes mellitus 1 and 2 කියලා තියෙනවානේ. මේක function වෙන්න පුළුවන් 1 ටද 2 ටද. Insulin release වෙන්නේ නැත්තේ මොකේද? Insulin release වෙන්නේ නැත්තේ මොකේද? Type 1 or type 2? Type 1 or type 2? Insulin release වෙන්නේ නැත්තේ. Insulin release වෙන්නේ නැත්තේ මොකේද? Type 1 ද type 2 ද? Type 1 ද type 2 ද? Yeah, right. So type 1 is called the insulin dependent diabetes mellitus and type 2 is called the insulin independent. එතකොට these nano devices are usually used to treat diabetes mellitus 1, type 1, where these devices can release insulin according to the blood glucose level. Okay, device it'll sense the change in blood glucose level, and according to the blood glucose level, if it's too high or above a certain value, they can release insulin. And if it decreases, then the insulin secretion is stopped. Okay. This device stores enough insulin. මට ඒක නම් ඇත්තටම sure නැහැ. Stores. It doesn't, I don't think it stores. Let me check that. Okay, it's something like this. Yeah, it does store. It does store. So it's like this. You give a method device again. I mean, in the internet, it wasn't about a device. It's basically these nanoparticles which encapsulate insulin. You can insulin ඇතුළට දාලා sphere එකක් වගේ හදපු nanoparticles are taken into your body via oral therapy, and they can be in the blood, or basically they are just present in the blood, right? So the insulin is encapsulated in the nanoparticle, right? So what happens is, according to the changes in the blood glucose level, this insulin is released. So okay, එකක් නෙමෙයි, sometimes it might be ගොඩක් දාලා තියෙනවා, but only the required amount is released to regulate the amount of insulin release. ඒ කියන්නේ හිතන්න 5ක් තිබ්බ කියලා. ඒක කැතයි. Just imagine there are 5 nanoparticles. 5 nanoparticles, and the blood glucose level increased only slightly. එතකොට only one might release the insulin, not all 4. Right? Only one might release the insulin. So these nanoparticles have insulin stored. It's like they have small concentrate. They are like packets of insulin. They are like packets of insulin. Unnamed there, right? They are like packets of insulin. And according to the blood glucose level changes, these packets are broken and the insulin is released and it decreases the blood glucose level, right? That's what happens. Okay, right, right. There has been successful applications of nanotechnology to the treatment and management of pain, both in clinical and experimental studies, like the fabrication of nanoformulated liposomes to the delivery of drugs for pain therapy, right? So, මේක නම් කරන්නේ pain therapy නම් සාමාන්‍යයෙන් කරන්නේ they now in your body you get these pain receptors නේ, right? In your body you get the pain receptors. So, pain therapy කියන්නේ basically the pain receptors are like මොකද ඒ කියන්නේ අර ඒව නිකන් හිරිවට්ටනවා, right? The pain receptors are understimulated. Oh, අපි ඒකට වචනයක් කියනවා මම කියන්න දෙන්න. But they are හිරිවට්ටනවා වගේ, right? So these liposomes can basically it's like a nanoparticle made up of a lipid membrane, right? Liposomes කියන්නේ it's a nanoparticle made up of lipid membrane, right? Lipid membrane which involves in this pain therapy, right? So 8, 9, this is the 10th one. Since nanocarrier systems can be easily transferred to the airways, many respiratory diseases can be treated using nanotechnology, right? So nanocarrier systems, that's the 11th one. Basically, they are just given into you, just given into the inhaled air. And these nanocarrier systems can basically go in search of these small infectious particles and destroy them in the respiratory system. Then Viva gel is the last one, I think. Last, the last. So Viva gel is a vaginally applied microbicide, right? So it's a microbicide which prevents the infection of HIV and HSV. HSV කියන්නේ herpes simplex virus. So ඒක pneumonic එක මතක තියාගන්න. HSV human sorry herpes simplex virus. HIV කියන්නේ මොකද්ද? What's HIV? What's HIV? What's HIV? HIV කියන්නේ මොකක්ද දන්නවද? එතකොට තමයි type කරනවා. HIV කියන්නේ මොකක්ද කියලා මේ AIDS කියන්න එපාලු. HIV is the virus, AIDS is the disease, right? AIDS is the disease, HIV is the virus which causes AIDS. So human immunodeficiency virus, right? Human immunodeficiency virus is HIV. HSV is herpes simplex virus, right? So this Viva gel, it's a microbicide එකක්. So it will kill these HIV virus particles and HSV virus particles, and it prevents this infection or spread of these STDs, right? It prevents the spread of these STDs. Viva gel is a product of nanotechnology which prevents the entry of HIV and HSV during sexual intercourse, right? So that's the 12th one. And the 13th one is nanocomposites. Nanocomposites are to replace broken bones and fill teeth as well. So it's something like cement. Right? Nanocomposites are very something like cement, where they are used to fill your broken teeth and broken bones. Right? So that's the last main function. Right? So that's the last main application. So was that clear? Clear now about these nanotechnologies and all that? Is it clear? Nanocomposites. No, it's not. It's not nanocomposites. It's nanocomposites. I think that might be a typing error. Yeah, it's nanocomposites. Nanocomposites නෙමෙයි. Okay, it's nanocomposites. Just make that error. එක මම දැක්කේ දැන්. Nanocomposites. Okay, මෙතන තියෙන nanocomposites කියලානේ. It should be nanocomposites. Okay? Right. Right, okay, let's quickly go through the applications again. So titanium dioxide and silver. Titanium dioxide and silver are basically sterilization nanoparticles, right? So they are involved in sterilizing hospital theaters or operation theaters and surgical instruments. Then also they can destroy the microbes. So that's anyway why they act as these sterilization nanoparticles. Ne? Then silver nanolotions are applied in the theaters to prevent the entry of microbes, and then you get titanium dioxide and silver nanofilters as well, which are used to observe or examine the SARS patients, right? SARS patients. Then you get nano device sensors which can detect, which are injected in your body into your body and can detect these changes in blood pressure, blood oxygen levels, and hormone concentrations. Then you get nanoparticles which can unclog blood vessels, and also some nanoparticles can eliminate cancer cells. Basically, an example is the gold nanoparticles. Gold nanoparticles can go in search of these specific cancer cells and destroy those specific cancer cells only, where they would release a certain chemical cargo only if they found the cancer cells, and that would reduce the side effects highly. Then you have these nanoshells. So nanoshells are spherical nanoparticles which consist of a dielectric core, and gold nanoshells are important for bioimaging enhancements. So ඔයගොල්ලන් අහුවොත් question එකක්, state two functions of nanoshells. What would you write? Structure එකට question එකක් එනවා. State two functions of nanoshells. What would you write? State two functions of nanoshells. What would you write? State two functions of nanoshells. What would you write? Yes, yes, I want everyone to answer. Good. Yes, right. Involved in treatment of cancers, involved in the treatment of cancers and bioimaging enhancements as well, right? Treatment of cancers and bioimaging enhancements as well. So, what are the functions? Two main functions of nanoshells. Then you get these nano devices which can treat diabetic patients, where they can release the required dose of insulin at required time intervals. And you get nanoformulated liposomes which can deliver drugs for pain therapy. Then you get the nano carrier systems that can easily transit to airways and treat the respiratory disorders. Vivagel, a vaginally applied microbicide which can prevent the entering of the HIV virus and HSV virus during sexual intercourse. And nanocomposites to replace the broken bones and to fill the teeth, right? So it's something like a cement. Okay, so that's it about nano technology. Let me see what questions I have. Right. Try this question. You send me an answer. It's a very easy question, not that difficult. Right. Okay. Good. That's easy, ne? Say the magic statement, nanocomposites, fill teeth, yes, fill teeth and broken bones. Nano carrier systems, unclog blocked arteries and eliminate cancer cells, NEME. That would be treatment of respiratory disorders, right? Respiratory disorders. Nano device sensors, bioimage enhancements, NEME. They are able to uh, detect these blood pressure, blood oxygen levels, and hormone concentrations. Gold nanoshells treatment of cancer. Gold nanoshells, name right? Gold nanoparticles are involved in the treatment of cancer, right? Not gold nanoshells. Gold nanoshells karani bioimaging enhancement. Yeah, bioimaging enhancements. Then Vivager prevent the entry of Neisseria gonorrhea enemy, prevents the entry of HIV and HSV. Right, try this question. Right, quickly. That's easy, right? Yes, right. The hospital is implementing new sterilization protocols for its operation theaters and surgical instruments, which of the following particles of pairs of nanoparticles would be most effective for this purpose. Then it's of course the titanium dioxide and silver nanoparticles, right? Titanium dioxide and silver nanoparticles. That's easy. No, these are not possible questions. Okay, these are not possible questions. Nanotechnology winning past paper structured negative, not as an MCQ. But I think 25 well MCQ එන්න ලොකු chance එකක් තියෙනවා nanotechnology වලින්. I feel like something might come. Try this. Right. Imagine a scenario where a patient with severely blocked arteries requires an uninvasive treatment. Which application of nanotechnology offers the most direct and precise solution for this condition? That's a good question, eh? Administration of smart drugs to target specific cells? No, it's a blockage in the arteries, so specific cells target can no, no. That would be for cancer treatment. Use of nanofilters to prevent viral infections? No, again, it's not related to this. It does prevent viral infections, but not related to this. Deployment of nanoparticles to unclog blocked arteries? Yes, ne? That would be the best. That would be the best. So third one is the answer. Implementation of nano device sensors to monitor hormone levels. That's again not related to this. Application of gold nanoshells for bioimage enhancements. Again, not related to this. Right? Not related to this. Right. Right. Try this question. Right. What tiny nanoparticles are used in smart drugs developed with nanotechnology for cancer treatment, and what key benefit do they provide in delivering treatment? Right. Titanium dioxide, silver nanoparticles, both of them are not smart drugs. They are just sterilization tools, right? Not smart drugs. ඒ අපි ගන්න නැහැ නේද ඒකට? අයන් ඔක්සයිඩ් කියලා එකක් කොහොමත් නැහැ. කාබන් නැනෝ ටියුබ්ස් කියන එක නෑ. මේ කාබන් නැනෝ ටියුබ්ස් කියන එක නෑ. ඔක එන්නේ ඇත්තටම නැනෝ කොම්පොසයිස් යටතේ. But you don't get that anyway. Gold particles seek out and destroy damaged cells rapidly and effectively with a very few side effects. Right? That would be the best answer. That would be the best answer. So third one is the answer. Right. Is it clear? Right. So again, the try to remember the applications, right? Try to remember the applications. Right? Try to remember the applications, and that will be really helpful. Okay. Right. So in the 2022 structured essay, what have they asked? They have asked what are known as nanoshells, what are known as nanoshells, and say two uses of nanoshells. So that was a structured essay question in 2022. So what are nanoshells? Spherical nanoparticles consisting of dielectric core are called nanoshells, and the two uses are treatment of cancer and bioimaging enhancements. So I think we're හරිම ලේසියිනේ. මට ඔය වගේ දේවල් ඉතින් ගොඩක් අතාරිනවා. ඉතින් කරලා නැහැ පාඩම. Right? So just go through this. Right. Then let's go to stem cell therapy. Right. Stem cell therapy gathot. Right. Stem cell therapy gathot. Stem cells. Now, in plants, you have meristematic cells. Right. In plants, you have meristematic cells. The plants are meristematic cells. And in humans, we have the stem cells. Okay. So basically, meristematic cells can plants. What happens to meristematic cells? Meristematic cells they anyway divide, right? Meristematic cells they divide, right? They divide and form more meristematic cells, and some others will be differentiated into other cell types, right? So meristematic cells, they constantly divide anyway, while some of them will remain as meristematic cells and others are constantly differentiated into other cells. So again, in our body, we have the stem cells. Meristematic cells, but in our body, we have the stem cells, which basically form more stem cells. They form more stem cells, while the rest of the stem cells, දැන් හිතන්න එක stem cell එකකින් 10ක් දුන්නා කියලා, 10 more stem cells. So if 2 of them remain as stem cells, the other 8 will differentiate into different cell types. Right? So basically, these stem cells are always constantly undergoing mitosis, right? Constantly undergoing mitosis in our body, and they are located at different stages, right? So you get the definition from that, where stem cells are undifferentiated cells which give rise to cells of the same type, right? So stem cells give rise to stem cells, okay? Meristematic cells give rise to meristematic cells, but some cells of the stem cells will differentiate. Stem cells are then stem cells, but some cells will differentiate as well. Some will remain as stem cells, right? So they can divide by mitosis without a limit, or at least for the lifetime of the animal. Again, after the animal is dead, anyway, you don't get these stem cells dividing. They are capable of terminally differentiating into other cell types. So they are capable of terminally differentiating into other cell types. So you get stem cells which produce more stem cells, and some of them remain as stem cells while others are differentiated into other cell types. So what is the process that these stem cells undergo? That's definitely mitosis. Okay. So basically, stem cells only when එකක් තමයි for our growth, right? Anyway, you need to grow. So your red blood cells or your skin epithelium, or basically the sperm cells, the ovaries, whatever develops initially from these stem cells, right? So stem cells එක වැඩ කරන තව cell જાතියක් කියනවා, we call them mesenchymal cells. Mesenchymal cells කියලා කියන්නේ යාට. ඒක අපිට අදාළ නැහැ දැන්. But these stem cells are very important for our growth, and it's very important for regeneration or replacement. If we are injured, what replaces our injuries and damaged tissue is basically these stem cells. Okay, so stem cells are basically very important for our growth. We need stem cells to grow, or else we will not grow, and it's very important to replace the tissues which are damaged as well, right? Do they constantly differentiate? Some stem cells do constantly differentiate, while some stem cells do not. Some are differentiate constantly, some are constantly differentiate. That it depends on the type of stem cells, right? So stem cells are required whenever there is a recurring need to replace differentiated cells that cannot themselves divide. If your skin epithelium is damaged, these epithelium cells are already differentiated, right? They are already differentiated. So they will not divide and replace the damaged tissue. What replaces the damaged tissue? When doing the skin, you get the epidermis. You get the epidermis and then the dermis. This is the dermis, this is the epidermis. What is found at the lowermost level of the epidermis? At the lowermost level of the epidermis. What do you get at the lowermost level of the epidermis? Yes, lowermost level of the epidermis. What do you get? Yes. Yes, right? The germinative layer, right? The germinative layer. Okay. So you get the germinative epithelial layer. And you get the stem cells, right? So if this area is damaged, what happens is these germinative epithelium layer will start producing more and more epithelial cells, which they will start actually producing more stem cells. These stem cells produce more stem cells where they will differentiate into the epithelial cells. Okay, they will start differentiating into the epithelial cells. So that's what actually happens. Okay, that's what actually happens, right? So a germinative epithelium or germinative layer, you get this germinative epithelium where they get this, they have the stem cells and they are dividing to form more stem cells which differentiate into epithelial cells. Okay. So if you need to replace the differentiated cells, you need the stem cells. So the stem cell itself has to be able to divide, but it doesn't necessarily have to divide rapidly. In fact, stem cells usually divide at a relatively slow rate. So it doesn't actually divide very rapidly. IN divide when just at a slow, normal rate it divides. Okay. Right. So stem cells කියන්නේ මොනවද කියන එක ගැන idea එකක් ආවද? What are stem cells කියන එකෙන් idea එකක් ආවද? What are stem cells කියන එක idea එකක් ආවද? Where about the others? Right. So stem cells are basically how all our cells are developed, right? Stem cells is like the basic stage of a cell where it needs to develop and undergoes differentiating into these different cell types that we have today, right? So the epithelium cells have their own stem cell line where those stem cells produce epithelium cells, and in our bone marrow there's something called the hemopoietic stem cells which develop into the blood cells, right? So you have these stem cells which needs to constantly divide into these new cell types. Can't differentiated cells divide? No, actually, usually differentiated cells. Usually, monkey and ever here, but usually differentiated cells do not divide. Okay, the epithelium cell that's a differentiated cell. It will not divide. What divides is the stem cells. Okay, red blood cell it doesn't divide. What divides is the stem cell to produce more red blood cells. Okay, so differentiated cells in our body actually do not divide. They enter that G0 stage. A cell cycle G0 stage. When doing the cell cycle, you learned most of the cells are actually in the G0 stage. But these stem cells, they do not enter the G0 stage. They can always divide, right? They can always definitely divide, right? Hare. So now we come to the most important part. So when we consider stem cells, there are two main types, right? Two main types. We call them embryonic stem cells and embryonic stem cells and adult stem cells, right? So when we consider embryonic stem cells, it's like this. So now a sperm nucleus and an egg nucleus fuses during process called fertilization, and that process results in a zygote. Zygote. What is the stage after the zygote? 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 Fertilization, zygote has ඊට පස්සේ zygote की මොකද දෙන්නේ? Yes, right. It's the morula, right? Morula. Morulla නෙමෙයි morulla කියලා එක්කින්නේ නෑ. Morula. Right? Morula. Morula. මේ morula කියලා තමයි සාමාන්‍ය කියන්නේ. Right? So morula is formed first. Then from the morula what's produced? From the morula you get? From the morula you get? What do you get from the morula? You get the blastocyst, right? Blastocyst. Blastocyst, right? You get the blastocyst. So from the morula, you get something called the blastocyst. Okay. So blastocyst is formed basically 5 to 7 days after fertilization. 5 to 7 days after fertilization, you get the blastocyst, right? So then this blastocyst, now blastocyst has something like this. It has something called the outer cell mass and the inner cell mass. So basically, I begin to make it a trophoblast and the inner cell mass. So the trophoblast is basically like a membrane, right? It's like a membrane, and in the middle of this, or middle can in this trophoblast, you get something called the inner cell mass. Okay, you get something called the inner cell mass. So මේ එළිය තියෙන මොකද්ද? දැන් blastocyst එකේ parts ටික කියන්නේ. right? මේ එළිය තියෙන මොකද්ද? We call this the outer cell mass, and the inner portion is called the inner cell mass. And later on, we call this the trophoblast, and this is called the embryoblast. And when going to medicine, you will learn that this trophoblast forms two more called the syncytial trophoblast and the cytotrophoblast, and the embryoblast forms two more called the epiblast and the hypoblast. So anyway, this trophoblast is what forms the placental portion of the fetus, right? Trophoblast is the membrane. So this is like what forms the placental portion. Chorion එක එහෙම හදන්නේ. දැනන්නේ chorionic වෙලා එහෙම. Chorion එක එහෙම එන්නේ මෙයාගෙන්. So this like forms the placental portion, right? Placental portion of the fetus. Embryoblastic can what? Embryoblast is actually the embryo which develops into the fetus, right? It develops into the fetus, right? So you get the blastocyst. Blastocyst has an outer cell mass and an inner cell mass. Then this outer cell mass becomes a trophoblast, which actually forms the placental portion of the fetus. So chorion name ඔක්කොම එන්නේ trophoblast එකෙන්. Then the embryoblast. Embryoblast ගත්තොත් that is what develops into the fetus. So even us, we were an embryoblast at a certain day. Okay, we were an embryoblast. So this embryoblast is the source of embryonic stem cells. These group of cells developed into us, right? This small group of cells is what developed into us. When the body group that is what divided and formed us, right? So basically, this is what you call the embryonic stem cells, where the embryonic stem cells can form any cell type in our body, right? Then like what happens is when the zygote is formed, zygote undergoes mitosis to form the morula. Then morula it undergoes more mitosis to form the blastocyst, and the identical group of cells will form the outer cell mass and the inner cell mass. The inner cell mass, it has a group of inner cell mass inner cell mass has a group of identical cell types. It has a group of identical cell types. This inner cell mass, what it does is it starts dividing and differentiating into who we are today. So basically, each and every cell of that inner cell mass can form any cell type in our body, right? In our body, we have over 200 cell types. Okay. So according to the signals, or according to the genes present, according to the nucleotide change, according to the genes present, according to the signals, according to the chemicals present, these cells will even though they can differentiate into all the 200 types, මේගොල්ලෝ අර මේ දැන් ඔතනින් එක cell group එකක්, they will become the cardiac muscle cells. Another group of cells will become the skeletal muscle cells. Another group of cells will become the blood cells. So according to the differences in nucleotide sequences and chemical signals, they will differentiate into different cell types. But every cell type of the inner cell mass, which we call the embryonic stem cells, have the potential to differentiate into any cell type. Okay, so if a person extracts the embryonic stem cells, if a person doesn't have an arm, if a person doesn't have an arm, if another person extracts the embryonic stem cells, they can basically take these embryonic stem cells out and culture them. Right? Culture කරනවා කියන්නේ මොකද්ද? In the laboratory, these embryonic stem cells are grown. Right? ඒ කියන්නේ ඕවා අරගත්තාම දැන් මම කියවනේ stem cells form stem cells. Okay? Stem cells will divide to form the stem cells. Then according to the different chemicals present and the different DNA sequences, they will divide into these different types. හරිද? Stem cells will form stem cells. Then according to the different chemicals and different signals only, they will differentiate into the different cell types. හරිද? So you can take this inner cell mass, culture it in the laboratory to produce more cells, to produce more stem cells, and you can develop any type of cell out of that embryonic stem cells. So basically, you can basically regrow a complete arm by reprogramming or by programming or giving the necessary conditions for these embryonic stem cells to develop into an arm. If the person doesn't have a heart or if the person lacks cardiac muscle cells, you can program these cells to develop into cardiac muscle cells, right? So embryonic stem cells can develop into any type of cell. Is that clear? Is that clear? Was that clear? ඒක තේරුණාද? Right, but the only issue is basically, isn't it like a stem cell? Yes, it's like a stem cell. That's what I said. This is called embryonic stem cells, right? Embryonic stem cells can, because the source is an embryo. The source is an early embryo. So you can make ethical issues, right? Ethical issues. We call these stem cells, these embryonic stem cells can basically develop into any cell type. They can develop into any cell type, right? Any cell type. So stem cells, we call them pluripotent stem cells, right? Pluripotent stem cells can, out of those stem cells, different types of cells can be produced, right? Different types of cells can be produced. That's what you call pluripotent stem cells. So embryonic stem cells are pluripotent stem cells, but they are not extracted. The source of these cells is an early embryo. So basically, you have to extract a developing child to get these cells, right? So ඒක කරන්නේ නැහැ usually because it's unethical, but we'll see what they do later. Right, බලමුකෝ කියවලා. So two cell types, embryonic stem cells, adult stem cells. Embryonic stem cells, following fertilization of a sperm and an egg cell, the fertilized egg is called a zygote. The zygote divides rapidly. In humans, around 5 to 7 days after fertilization, the dividing cells create an embryo consisting of a small hollow cluster of approximately 100 cells called the blastocyst, right? So the blastocyst is 100 cells. The blastocyst is approximately a 1/7 of a millimeter in diameter. Again, 1/7 of a millimeter, right? So you get a blastocyst which is about 100 cells, and it's about, the diameter is about 1/7 of a millimeter. There is a small cluster of around 30 cells tucked inside the blastocyst that forms a structure known as the inner cell mass. So out of these 100 cells, about 30 forms the inner cell mass. These cells are the source of embryonic stem cells. Embryonic stem cells are so special because they can eventually differentiate into all forms, differentiate to form all of more than 200 cell types that make up the human body. Anything embryo එකකින් හැදෙන්නේ this human body. So any cell out of that embryonic stem cell can differentiate into any of the 200 cell types in the body. Therefore, embryonic stem cells are called pluripotent because they have the potential to develop into a variety of different cell types. So they might ask you, why are embryonic stem cells called pluripotent? If they ask you why are embryonic cells called pluripotent, that's because they can develop, they have the potential to develop into a variety of different cell types. That's what you call pluripotent. Okay. The human embryonic stem cells are unspecialized cells with two major functions. So the embryonic stem cells have two major properties. එකක් තමයි, they can self-renew indefinitely, produce more stem cells. මං කිව්වේ these stem cells can produce more stem cells indefinitely. Ours produce කරගෙන යන්න පුළුවන් ඒගොල්ලන්ට. The division is not very fast, but still they can produce more stem cells. And the other is under the proper growth conditions. These proper chemicals and proper signals are needed. So again, these stem cells will develop into stem cells will develop into cardiac muscle cells only according to signal type 1. The signal and chemical type 2 are what? It develops into skeletal muscles. If another signal is given, type 3 signal, they develop into blood. Right? So according to the different conditions, they change what cell type they differentiate into. So under the proper growth conditions, human embryonic stem cells can differentiate into a variety of mature cells with specialized functions. So embryonic stem cells embryonic stem cells embryonic stem cells embryonic stem cells right. Embryonic stem cells can differentiate into cell types, so we can control the conditions within. When we change the growth conditions, according to the growth condition, they will mature into different cell types. So we can change the growth conditions. Even during tissue culture, according to the concentrations of cytokinins and auxins, they will develop into the shoot or the root. And again, we can alter the growth conditions and develop them into different cell types. Right, then adult stem cells. So adult stem cells can basically, we in our body have adult stem cells, right? We in our body have adult stem cells and they can develop into different cell types. They only form a specific type of cell. And they will form only a specific type of cell. Again, in your bone marrow. In your bone marrow, you get hemopoietic stem cells, right? You get hemopoietic stem cells. They will give rise only to blood cells, right? They will give rise only to blood cells. Then in your in your intestine, you get intestinal stem cells, right? Intestinal stem cells. They will give rise only to intestinal epithelium, right? So again, there are many different types of adult stem cells, right? There are many different types of adult stem cells, and these different types of adult stem cells can give rise to a specific type of cell. Okay, so that's about adult stem cells. So adult stem cells are found throughout the body after development. Adult stem cells are present in many tissues. There are many types. There are many types of adult stem cells specialized for the genesis of different classes of terminally differentiated cells, and hence each type of stem cell serves for the renewal of only one particular type of tissue. Hemopoietic stem cells now only the blood tissue. Intestinal stem cells now only the intestinal epithelium. Okay. So examples within epidermal stem cells now only for the epidermis. Intestinal stem cells now only for the intestinal epithelium. And hemopoietic stem cells only for blood. Neural stem cells only for the central nervous system. Okay, so one stem cell type only forms a certain type of cell. Stem cells can be isolated and cultured in culture media in vitro. When adult stem cells are removed from the body and maintained in culture or are transplanted from one side of the body to the another, they generally remain faithful to their origins. Again, they generally remain faithful to their origins. Now, embryonic stem cells, you can program them to form any type of cell. But if you take hemopoietic stem cells and put them into your damaged epidermis, then for the regeneration of your epidermis, you need epidermal stem cells. Okay, for the regeneration of your epidermis, you need epidermal stem cells. But you extract hemopoietic stem cells from the bone marrow and put it to the damaged area in the epidermal stem cells. The epidermis is not produced. IE, these hemolytic stem cells will only produce blood cells, only produce blood tissue. You know, only red blood cells or white blood cells or platelets are produced. Yeah, that's what they mean by they generally remain faithful to their origins. Okay. This creates practical limitations of using adult stem cell for tissue regeneration. In contrast, embryonic stem cells can be kept proliferating indefinitely in culture and yet retain the unrestricted development potential. In culture, basically laboratory conditions, these embryonic stem cells can be extracted and they can be kept dividing. Right? They using just a small number of embryonic stem cells, you can produce a large number of embryonic stem cells because they are constantly dividing. And from those embryonic stem cells, you can develop different types of tissues as well. But adult stem cells වලින් එක ජාතියකින් එක tissue ජාතියක් produce කරන්න පුළුවන්. If embryonic stem cells are put back into the blastocyst, right? So embryonic extract කරපු embryonic stem cells, if they are put back into the blastocyst, they become incorporated into the embryo and can give rise to all the tissues and the cell types in the body. So basically, if you put them back into the blastocyst, they will give rise to a complete organism, right? They will give rise to a complete organism. Embryonic stem cells are in blastocyst sac at them, you can produce a complete organism from that as well. Under the proper growth, including germ cells, under the proper growth conditions, human embryonic stem cells can differentiate into a variety of mature cells with specialized functions in vitro. There are ethical concerns of obtaining human embryonic stem cells. Therefore, research on HSCs is very controversial because of their source, an early embryo. Right? So the source is an early embryo. Basically, specifically, the source of embryonic stem cells is the inner cell mass of the blastocyst. Right? The inner cell mass of the blastocyst. Right. But the thing is, then embryo can extract and ban embryonic stem cells because that's very unethical. You are extracting stem cells from a developing baby. That's very, very unethical, right? And it's very controversial. So what the scientists did was they took adult stem cells, right? They took adult stem cells, and they genetically altered these adult stem cells, right? They genetically altered these adult stem cells. Genetically altered these adult stem cells and reprogram them so that they become embryonic stem cells or that they can produce a number of different stem cells. This is called, then අපි ගත්තනේ මේගොල්ලන්ට කිව්ව නමක් අපි නමක් කිව්වා pluripotent stem cells, right? Embryonic stem cells are called pluripotent stem cells because they can differentiate into a large variety of cell types. So may adult stem cells are again, they genetically modified them and altered them to form a pluripotent stem cell, right? Adult stem cells are again pluripotent stem cell like හැදුවා. So we call these does it affect the baby's health and extract stem cells? No. You don't extract stem cells from babies, okay? You don't extract stem cells from babies. You extract stem cells from the inner cell mass, okay? Not from a baby. From a developed baby, you cannot extract embryonic stem cells. You need to extract it while at the stage of inner cell mass. Okay? No, it hasn't developed into a fetus yet. It hasn't developed into a fetus yet. Fetus එකක් කියන්නේ differentiate වෙන්න පටන් ගන්නවා. Right? The embryonic stem cells start differentiating. What if it's like IVF? What do you mean? What if it's like IVF? In vitro fertilization? What do you mean? එහෙම ගන්න පුළුවන්. But still, no, yeah, it's still unethical. You take a sperm and an egg cell from a person and you join them together to form an embryo which has a potential of developing into a certain human, and you extract these embryonic stem cells from a potential human. I think that's unethical. Even though it's in the laboratory condition, that's still unethical, okay? I mean, you can carry out a secret laboratory experiments after you finish A levels and get into universities, but usually it's considered unethical. ඕන්න ලොකු වෙලා ඉතින් කරාට කමක් නැහැ. ඉතින් ප්‍රශ්නයක් නැහැ. අහුවෙන්න එපා එච්චර. Right. So දැන් අතන එහෙම කරන්න ඕනේ නැහැ. Right? එහෙම කරන්න ඕනේ නැහැ. There's no need to extract them. That's what I'm telling. ඒක extract කරන්න උවමනාවක් නැහැ දැන්. Is it illegal? It is illegal ඉතින් නේ. It's not It's not ethical, it's illegal thing, of course. Like illegal. And I don't think they have permission to do that. Right? නෑ එහෙම කරන්න උමනාවක් නැහැ. ඒලා තමයි කියන්න හදන්නේ. So embryonic stem cells extract කරන්න උමනාවක් නැහැ දැන්. ඇයි these adult stem cells are genetically altered where they are programmed or reprogrammed to produce any cell type. We call these induced pluripotent stem cells. Right? Induced pluripotent. හරි. Pluripotent stem cells කියන්නේ මොනවාද? Those are the stem cells which can develop into a variety of cell types. එහෙනම් in the adult stem cells, this pluripotent nature is induced, right? It's induced. The pluripotent nature is induced in these adult stem cells. Okay, so that's what you call induced pluripotent stem cells, right? This pluripotent nature is induced in them. Then you can produce different cell types from these induced pluripotent stem cells. Okay, so that's what they say here. In late 2006, scientists demonstrated the successful derivation of pluripotent stem cells from adult tissues by reprogramming human cells taken directly from a volunteer. Okay, these are known as induced pluripotent stem cells. Pluripotent stem cells hold promise in the field of regenerative medicine because they can be propagated indefinitely as well as give rise to every other cell type in the body, such as neurons, heart, pancreatic, and liver cells. They represent a single source of cells that could be used to replace those lost due to damage and disease. Right? Again, if a certain person lost his or her arm, using these induced pluripotent stem cells, that whole arm can be regenerated because these adult stem cells are reprogrammed um, uh, 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 to produce these induced pluripotent stem cells where they can differentiate into different cell types. Okay, so it's a very important milestone in regenerative medicine. Okay, since induced pluripotent stem cells can be derived directly from adult tissues, they not only bypass the need for embryos but can be made into a patient matched manner. So embryos are not needed anymore. Okay, so don't worry, you don't have to secretly harvest embryos. But I think if you can't, or if you don't have the technology to develop these induced pluripotent stem cells, you will have to extract from embryos, right? So it not only bypasses the need for embryos, but also can be made in a patient match manner. Again, patient match manner කියන්නේ you can take cells from a patient and produce his own set of induced pluripotent stem cells. Again, එයාගේ cells අරගෙන you can reprogram them into his own pluripotent stem cell line, where his own cells are used to regenerate his own body parts, which means that each individual could have their own pluripotent stem cell line. ඔයාට ඔයාගේ cell එකක් අරගෙන reprogram කරනවා into your own pluripotent stem cell line. මගෙන් අරගෙන මගේ cells reprogram කරනවා into my own pluripotent stem cell line. Right? How to reprogram? Reprogram can basically genes මාරු කරනවා. Reprogramming means you take a cell, change the genes, add some genes, and it reprograms. What if a person cannot harvest their own embryonic stem cells? Basically, embryonic stem cells තියෙන්නේ only in the blastocyst stage. හරිද? Blastocyst stage එක ඉවරනාට පස්සේ you don't get embryonic stem cells. You get adult stem cells only. Right? So a person cannot harvest his own embryonic stem cells, right? Because that person doesn't have embryonic stem cells. Embryonic stem cells තියෙන්නේ only at the blastocyst stage. එතනින් පස්සේ නෑ embryonic stem cells because the organism is developed, right? The cells are differentiated. Then you get adult stem cells only. හරිද? තේරුණාද? A pregnant person can give consent for their the pregnant person cannot give consent for the embryonic stem cells to be harvested because the pregnant person main embryonic stem cells, it's the embryonic stem cells of another organism. That you are telling something like this. The mother can give permission to harvest the stem cells of her own fetus. That's what you're telling me, right? So that's even unethical. The mother is asking them to harvest the stem cells of their own baby. Yeah, it's an organism after the fetal stage, but it's supposed to develop into a human, right? It's supposed to develop into a human. එතකොට ඉතින් ගන්න එක හරි නෑ නේ ටිකක්. මම කියන්නේ නිකන් සෙට් නෑ වගේ නේද? Maybe uh, you can grow up and give up your own embryonic stem cells. කිසි ප්‍රශ්නයක් නෑ. Can't be half without damaging the embryo. You are basically taking the cells away from the embryo. You are taking the cells from the embryo, right? You are just taking the cells from the embryo. I think anyway you are damaging the embryo. How can stem cells be harvested ethically? Embryonic stem cells අතර ethically harvest කරන්න බෑ. Adult stem cells ගන්න පුළුවන්. Because now when I consider them, well, leukemia දන්නවනේ. You guys know leukemia නේ. Leukemia is basically blood cancer, right? Leukemia is blood cancer. හරිද? එතකොට basically එකකින් කියන්නේ your blood cells are not developed properly. So that is due to these hemopoietic stem cells. Hemopoietic stem cells are the stem cells that develop into the blood tissue. So the hemopoietic stem cells are what's affected, right? එතකොට your blood cells are not developed properly and then can basically hemopoietic stem cells වල cancer එකක්. එහෙම දැන් blood cancer කියන්නේ leukemia වලට. So your blood cells are not developed properly. So what you can do is you can take these hemopoietic stem cells from a donor. දැන් හිතන්න මගේ කවුරු හරි යාළුවෙක්ට තියෙනවා නම් leukemia, they can take hemopoietic stem cells from a healthy person like me, from my bone marrow, and replace his damaged, genetically mutated hemopoietic stem cells and put මගේ ටික එයාට දෙනවා. මගෙන් extra එක මගේ ඔක්කොම ගන්න නෑ. මගෙන් ටිකක් ගන්නවා because anyways there are so much stem cells in the adult body නේ. So you can take some stem cells from my body and put them into his. So ඒ වගේ ඒවා ethical, but taking it from an embryo is not ethical. Right. Applications of stem cells. There are many potential applications of stem cells, from growing healthy tissues to studying them to understand and treat birth defects to genetic manipulation of delivering genes in gene therapy approaches. Creating whole tissues in the laboratory. So you can create whole tissues in the laboratory using tissue engineering is another potential application for repairing tissues such as damaged heart muscles and damaged spinal organs. So there are small kids with holes in their heart that can be treated with this stem cell therapy, right? That holes can be covered with this stem cell therapy. Then blood stem cells, hemopoietic stem cells taken from the bone marrow. Taken from the bone marrow of a healthy immunological compatible donor can be used to replenish the bone marrow of patients with leukemia. So leukemia in patients like bone marrow replace by using these hemopoietic stem cells of me. Scientists believe that stem cell technologies will play key roles in developing treatments to diseases such as stroke, heart diseases, Parkinson's disease, Alzheimer's disease, diabetes, and many more. Then ගත්තොත් heart diseases sometimes because due to the death of cardiac muscles. Using these stem cells that died off or marriage cardiac muscles replace කරනවා. Right? Parkinson's disease, Alzheimer's disease but it's due to these neural pathways that affect dopamine or something, right? Illegally taking bone marrow of people used for this. No, basically bone marrow extraction that's legal, right? මගෙන් bone marrow එකකින් පොඩි blood cells එකක් ගත්තා කියලා ඒක donors ලා ඉන්න ඕනි තරම් to donate bone marrow cells. That's not illegal. That can be taken නිකන් bone marrow cells ටිකක් දෙන එක. It's like donating blood. මම මතක නැහැ කොහමද මේ Parkinson's disease and Alzheimer's disease හැදෙන්නේ මොකකින්ද කියලා නම්. කොහොම හරි it affects your neurons in the brain, right? It's due to your damage to your brain, right? So what happens is those damages can be replaced by these stem cells, right? එතකොට this Parkinson's disease and Alzheimer's disease can be treated. Diabetes ගත්තොත් these insulin cells are damaged, right? The beta cells which produce insulin is damaged. එතකොට මොකක්ද වෙන්නේ? When that happens, you can replace the beta cells with the stem cells. So stem cells දාලා beta cells ටිකක් හද වෙනවා. So diabetes can be treated, right? So using the stem cells, many diseases can be treated. හරිද? So is it clear? If the embryo was not able to survive, can the mother then consent to donating embryonic stem cells like fetuses are given? They can anything. They can need. I don't know if the embryo doesn't uh, survive, then anything they can donate. Neither. Yeah, I don't know. You guys will be horrible mothers, huh? I mean, most are girls, nee. Horrible mothers. Like you're trying to donate your baby, nee. Harvesting embryonic stem cells from a criminal. It's still unethical thing. Yeah, you can make your own embryonic stem cells and donate them. කිසි ප්‍රශ්නයක් නැහැ. වෙන කෙනෙක් ගකින් ගන්න එපා. ඕගොල්ලන්ගේ ඕනිම ඕනි ඕගොල්ලන්ගේ embryonic stem cells ටික දෙන්න. right? කිසි ප්‍රශ්නයක් නැහැ. වෙන කෙනකින් ගන්න එපා. Right, okay. And even here මේ gene therapy කියන්නේ මෙතන මේ delivering genes for gene therapy. ඒ කියන්නේ some babies and all that have these genetic disorders නේ. They have genetic disorders නේ. එතකොට දැන් genetic disorder එකක් එන්නේ ඇයි? Due to a mutation in the genetic code, right? Due to mutation in the DNA sequence. එතකොට මොකද්ද ගන්න පුළුවන්? දැන් අපි ගත්තොත් cystic fibrosis, right? Cystic fibrosis ගත්තොත් cystic fibrosis is due to this effect in the gene of the cystic fibrosis transmembrane regulator protein නේ. CFTR protein එකේ පොඩි issue එකක් නිසානේ. ඒ කියන්නේ අර gene mutation එක නිසානේ ඔක වෙන්නේ. So the cystic fibrosis a cells ටික අපිට replace කරන්න පුළුවන් from the stem cells which develop into new cells which do not have that genetic disease. එතකොට cystic fibrosis නැත්නම් ඔය sickle cell anemia වගේ diseases, those genetically affected or genetically mutated cells can be replaced by the stem cells where they develop into the new cells. එතකොට genetic diseases can also be treated, right? Right. So I hope that was clear. Let's see what we have. මුලින් බලමු past paper questions තියෙනවද කියලා. Yeah, we have one past paper question from 2021. Try this question. Remember answers? I got some. Some are correct, some are wrong. Remember? Right. Stem cells can give rise to cells of the same type. Yes, they can give rise to cells of the same type, right? These stem cells can give rise to stem cells. Can divide without a limit? Yes, they can. Are of three types? No, they are of two types. Adult stem cells and embryonic stem cells. Are undifferentiated cells? Yes, they are undifferentiated cells. Divide rapidly? No, they divide slowly. So ABD, first one is the answer. Right? So very easy, right? All right. Let's see some more questions. Right, try this. Okay, I think most of you got it. Ne? Which of the following best describes the fundamental biological characteristic of stem cells? They are highly specialized cells that divide quickly. Ne? They are undifferentiated cells that can divide without a limit and can develop into various other specialized cell types. Yes, that is the definition. Ne? These are cells that only exist in adult tissues. No, even embryos have them. They are cells that repair themselves but lose the ability to divide over time. No, that's wrong. They can divide indefinitely. They are differentiated stem cells that can only produce more cells of their exact same type. No, they can develop more cells or they can produce different types of stem cells. Right? They can develop different types of cells. So it's the second one. Try this question. All right, let's see. When comparing embryonic stem cells and adult stem cells for using growing new tissues, what is the key difference in their capabilities? Adult stem cells can turn into any cell type. No, not into any cell type, only a specific type. Where are the second? Embryonic stem cells can keep dividing and become any cell type, but adult stem cells usually only make cells for their own original tissue. Yes, that's correct. They remain faithful to their origin. Adult stem cells are easier to get and grow in lab than embryonic stem cells. No, that's wrong. Embryonic stem cells are much more easier. Embryonic stem cells cause more ethical problems, but adult stem cells have no problems. No problems can bear. Embryonic stem cells do have ethical problems, but adult stem cells no problems can bear. Right? Both embryonic stem cells and adult stem cells have the same ability to create all body tissues. No, not all body tissues. Okay, adult stem cells can't produce all body tissues, so it's the second one. Let's see one more question. This is easy, right? This is easy. Try this. It's easy, right? Adult stem cells are found throughout the body and are specialized. Which of the following accurately pairs an adult stem cell with the tissue it typically renews? Neural stem cells for intestinal epithelium neme, for cells of the central nervous system. Epidermal cells for not the epididymis, it's for the epidermis. Hemopoietic stem cells for the central nervous system neme, for blood. Intestinal stem cells for internal capsule neme, for the intestinal epithelium. So fifth one is the answer, hemopoietic stem cell for blood. Right. Let's quickly watch a small YouTube video on embryonic stem cells.
Following fertilization, the developing embryo contains a group of cells known as the inner cell mass that will eventually go on to form all tissues of the body. However, at this early stage of development, these cells have not yet committed to become a specific type of cell and are said to be in an undifferentiated state. If the cells of the inner cell mass are isolated, they can grow indefinitely in a plastic dish and maintain this undifferentiated state. These cells are known as embryonic stem cells, or ES cells. Like cells in the developing embryo, ES cells retain the ability to form any type of cell in the adult body. Thus, ES cells are said to be pluripotent. By studying the signals required to control how ES cells develop into specific...
The monkey, like, you can control the different signals that are used to develop these embryonic stem cells in different times. So by controlling those signals, you can manipulate these embryonic stem cells to develop it into the required cell type.
Specific cell types, scientists may be able to develop therapies capable of restoring damaged tissues.
That's about embryonic stem cells. We have one more about adult stem cells. Yeah.
Pluripotent stem cells are critically important to the work of the UC San Diego stem cell program. They allow the Motri lab to grow neurons with neurological defects, and Carl Wallen's lab to grow retinal cells with eye diseases so they can try to find cures. But how are they made? 2012 Nobel laureate Shinya Yamanaka discovered that by reactivating only 4 specific genes in adult cells, they would become.
So the adult stem cells are reprogrammed කියලා. ඒක කරන්නේ basically adult cells are given 4 new genes, right? They are given 4 new genes and they are reprogrammed to become pluripotent stem cells.
Pluripotent, which means they can be reprogrammed to make any tissue in the body, like the neural and retinal cells the Motri and Wallen labs use, as well as many other types of cells used to study or treat diseases. Alison Motri's lab can take cells from inside your mouth and provide them with the genes that Yamanaka discovered. The genes are introduced into the cells using viruses that can deliver the genes in a way similar to how a virus causes infection in a cell. The genes cause the cells to produce specific proteins. These proteins enter the nucleus and act on the cell's DNA. This causes the cell's DNA to convert the cells into pluripotent stem cells.
So when those 4 type of genes are put into the adult stem cells, they are reprogrammed into the pluripotent stem cells.
These are called induced pluripotent stem cells.
A pluripotency induced in නිසා they are called iPSCs.
Or iPSCs. The lab then carefully cultures the iPSCs, which grow in a thin layer, constantly caring for them to make sure they grow well. One reason iPSCs are so useful and important is that since they come from adult cells, they carry the genetics of the donor.
That's why each every individual can make their own induced pluripotent stem cell line කියලා. You can take your own cells and create your own induced pluripotent stem cell line because those cells contain your genes, right?
Allows the Motri, Wallen, and other labs to capture the genomes of real disorders so they can work with the cells to find real cures, which the UC San Diego Stem Cell Program is doing right now.
Right. So were the stem cell therapy section clear? Was it clear? So in 2024 10th essay එකේ part C එකට ඇහුවේ applications of stem cells, right? Applications of stem cells. So the patient's immune system doesn't repel the iPSCs, no, right? The patient's immune system doesn't actually repel them because the iPSCs are made from your own cells. It's your own cells that they use, right? So your own cells the immune system doesn't repel them, right? Yeah, they're considered self cells. Okay. They're like your own cells right. Okay, then it's 9:02. We'll take a small break and come back and do the last section. It's not much, the Human Genome Project. ඒක ඇත්තටම කියන්න දෙයක් නෑ ලොකුවට. So let's take a small break, a 10-minute break till 9:12 and join again at 9:12 and we'll finish off the Human Genome Project and the whole lesson. Right? Right, okay. Let's continue then. The Human Genome Project, right? So, nothing very big to tell about this actually. So, the Human Genome Project, what it is actually basically in our body. Right? You guys can hear me, right? My Anani? The Tani given. Can you hear me? Hello. Okay. Hi. Okay. So the Human Genome Project is basically a project that was initiated in, I can't remember the year, 1997, right? 1997, the 19, no matter. Komari. It was a 13-year project initially, which was initiated so that all the genes in the human body can be decoded, right? Decoded. Now, when we consider our genes, the genes in our body, or basically humans can know the human species. You and me, 99.9% genes in our body are similar, right? 99.9% of the genes in our body are similar. So every human is similar to each other in a 99.9% manner. The only variation that we have is 0.1%. So we are different only in 0.1%. Okay, so every human is similar to each other in 99.9%. So when we consider the genes that are present in our body, we have about 20,000 protein coding genes, right? We have about 20,000 protein coding genes. And out of these 20,000 protein coding genes, so basically out of all the genes, 99.9% are similar to every human. Okay, 0.1 is different. Basically, our genes are even similar to that of chimpanzees, even mice, even elephants, right? We are, every organism is somewhat similar to each other at different percentages. We are similar to these chimpanzees and all that, 98% or something, right? Attack, okay, we are similar to the chimpanzees. So again, this Human Genome Project was initiated so that all these 20,000 genes could be decoded. Now, genes are made up of what? What are genes made up of? What are genes made up of? What are genes made up of? Yeah. What are genes made up of? Yes, what are genes made up of? Yes, right? Genes are made up of nucleotide sequences. So genes are made up of nucleotide sequences. Hey, now, these nucleotide sequence, if you know the sequence of the nucleotides and if you can uncode that sequence, basically you know what nucleotide sequence is making that gene. So again, this Human Genome Project was originally designed so that these nucleotide sequences of these 20,000 genes could be identified, right? So initially it was a 13-year project, but now you can basically go to there are the centers. But in foreign countries, there are the centers where you can go there and get your own genetic sequence in just about hours, right? You can get your own genetic sequence in just some hours, right? Kiel Balum. The human genome has been the focus of biological research for the last 2 decades and will continue to be the center of attention for many years to come. It is a worldwide effort to identify all the human genes of each chromosome, along with several other goals. The Human Genome Project is an enormous undertaking in genomics that is providing scientists with exciting insight into human genes, their locations and functions. So basically, in the Human Genome Project, other than uncoding the nucleotide sequences, their locations or the functions are also uncoded or identified. So what do we call the location of a gene? What do you call the location of a gene? What do you call the location of a gene? It's called a not an allele, it's called a locus, right? Allele are basically variations in a gene. Gene again, two different variations, right? The locus is where what you call a location of a gene, right? Location of a gene is a locus, or loci is a plural word. It was originally a 13-year project, 1990 to 2003. 1990 to 2003. 13-year project coordinated with the US Department of Energy and National Institute of Health. Additional contributions mainly came from UK, Japan, France, Germany, Australia, and China. So we're රටවල් ටිකෙන් දෙක තුනක් තියාගන්න. කොහොමද මතක තියාගන්න ඕනෙත් නැහැ. ඉතින් මේ මුන් ටික තමයි දෙක දන්නවනේ කොහොමද. Right? So it was initially in the US. Okay. So the aims of the project were to identify all the genes. So approximately 20,000 protein coding genes in the human genome. Determine the sequences of the 3 billion chemical base pairs that make up the human DNA. So these 20,000 protein coding genes are made up of 3 billion base pairs. So 3 billion base pairs involved in the formation of these 20,000 protein coding genes. Right? 3 billion can you 3 into 10 to the 9. Ne? Billion can 10 to the 9. Ne? Yeah, ne? Yeah, 10 to the 9. Ne? So you divide by the number of genes, 2 into 10 to the power 4. And you get basically per gene, how many base pairs? Per gene, you get about 1.5 into 10 to the power 5 base pairs per gene, right? Per gene, you get about a million genes, almost a million genes, right? Store information on databases. So these nucleotide sequences are stored in databases. Improve tools for data analysis. So improve the tools for these identification of genetic sequence. Again, as the genome project went on, first generation DNA sequencing technology, second generation DNA sequencing technology, third generation DNA sequencing, right? So the technology developed as the genome project was going on. So this process is actually called DNA sequencing. The DNA sequence or the nucleotide sequence for these genes are identified. Translated technologies to private sector address ethical, legal, and social issues that may arise from the project. So may hire පුළුවන් මතක තියාගන්න. Right? hire පුළුවන් මතක තියාගන්න. මොකද ඔය ඕක කොහොම මතක අපේ level අවුරුද්දේ. We'll see බලු. but පුළුවන් hire මතක තියාගන්න. A major quality assessment of the human genome sequence was published in 2004, indicating over 92% of sampling exceeded 99.99% accuracy, which was within the intended goal. Right, so මේක නැත්තන බොරු හැල්ලක් තියෙන්නේ කවන්න කියලා බලමුකෝ. The Genome Reference Consortium, which is an international collective of academic and research institutes with expertise in genome mapping, is still working on the analysis of the data obtained from the Human Genome Project. The sequencing of the human genome holds benefits in many fields. All right. So it holds benefits. So basically benefits, right? Benefits. The benefits are these, right? Benefits are these. So molecular medicine to human evolution. So improved diagnosis of various diseases. Identification mutations linked to different forms of cancer. Basically, you get these genetic disorders, right? You get genetic disorders. And so if you identify the genetic sequence that causes a certain genetic disease, you can basically treat it, right? You get a DNA sequence like this. You have a DNA sequence at and a certain disease is caused when these 3 nucleotides change. Okay, when these 3 nucleotides change. Change a certain disease is caused. So you take a normal person and compare it with this diseased person. Then you can identify which nucleotides change. You take a normal person and you take a diseased person, right? So you can identify which person is diseased. Which person is diseased. What you can do is now you know the gene sequence or the DNA sequence that was supposed to be in the person to make him healthy. In order to replace this DNA sequence, you add or bring in another gene, right? Molecular biology. Using a vector, you bring in the corrected gene. Corrected gene right? Hari gasabu gene which replaces this damaged gene. Genetic disorders can be cured, right? So human genome project important identification or diagnosis, right? Because you can diagnose the genetic disorder by analyzing this DNA sequence. DNA sequence, but you can identify it as this DNA genetic disease. And then you know that cancer cells are formed when a normal cell mutates into an abnormal cell, right? දන්නවනේ cancer cell වල formation එකේ පළවෙනිම step එක transformation කියලා. Transformation කියන්නේ මොකද්ද? A normal cell is transformed into an abnormal cell. Usually it occurs due to these mutations in the gene sequence. එතකොට if you can find the gene that causes the formation of cancer cells, then you can target that gene and destroy cancers. Basically in our body, මම එක જાතියක් කියන්න. In our body, we have these genes called tumor suppressor genes, right? In our body, our cells have something called tumor suppressor genes. Tumor suppressor genes. What do tumor suppressor genes do? They suppress tumors. ඒ කියන්නේ cancer cells form වෙන්න දෙන්නේ නැහැ. හරිද? But due to some disorders, these tumor suppressor genes might be inhibited. ඒවා inhibit වෙනවා. හරිද? එතකොට මොකක්ද වෙන්නේ? Tumors might be formed. So some cancers are due to these tumor suppressor genes being inhibited. එතකොට if you know the correct sequence of the tumor suppressor gene, basically you can put it into the cell and activate the tumor suppressor genes. You can activate can තව විදි තියෙනවා. But if you know the correct sequence of the tumor suppressor gene, you can put it into the cell and activate the tumor suppressor gene. Then what happens is the cancer cells are suppressed, right? So ඒ වගේ importances තියෙනවා. Then the design of medication and more accurate prediction of their effects. So basically, if your genome is sequenced, you can develop medicines according to your own DNA sequence. Again, mage DNA sequence එක අනුව medicines are produced to me. According to your DNA sequence, medicines are given for you. That is actually called personalized medicine, right? ඔයගොල්ලෝ අපි කියන personalized medicine කියලා. Right? Personalized medicine where according to your genome sequence, medicines are developed. And more accurate predictions of their effects. Gene therapy තමයි මම කියවර genes are inserted කියලා. That's gene therapy. And control systems of drugs. Study of human evolution. Again, using the genes, you can study human evolution and anthropology, right? Anthropology and human evolution can be studied using a genes. Another proposed benefit is the commercial development of genomics research related to DNA-based products. It's like a business, right? So DNA-based products develop කරන companies වලට important වෙනවා. The Human Genome Project and other genome projects, right? Now, this is very important. The other genome projects that are sequenced is E. coli, E. coli කියන්නේ Escherichia coli, yeast. Give another name for yeast. Another name for yeast. What's another name for yeast? What is another name for yeast? Another name for yeast. Saccharomyces නෙමෙයි. Saccharomyces කියලා එකක් නෑ හරිද? It's ඔයගොල්ලන් ස්පෙලිස් නම් වැරදි. ගොඩක් අයගේ it's Saccharomyces cerevisiae, right? Saccharomyces cerevisiae. Saccharomyces cerevisiae. Let me write it. So, I think this is it, right? Right, Saccharomyces cerevisiae. So the completed genome projects are E. coli, yeast is Saccharomyces cerevisiae, and mouse. Mouse, the mouse. And Arabidopsis thaliana, right? Arabidopsis thaliana, right? Arabidopsis thaliana and rice, right? So they have been completed. You need to remember these, right? Arabidopsis thaliana and rice. This has therefore opened the way to a comprehensive description of the molecular activities of human cells and the ways in which these activities are controlled. This is central to the continued development and not only in molecular biology and genetics, but also those areas of biochemistry, cell biology, and physiology now described as molecular life sciences. Right. Now this part is also important, quite interesting. So when you consider DNA, DNA ගත්තොත් මෙතන මේක DNA sequence එකක් කියලා. And if this is a gene, and if this is another gene, between the genes, what do you have? Between the genes, what do you have? This is a gene, right? This is a gene. Between the genes, what do you have? Molecular biology මතකයිද කියලා? Between the genes, what do you have? Between the genes, right? Genes, okay? Genes. Between genes, you have intergenic. Like in නමින්ම ගන්නකො ඉතින්. Genes දෙකක් මැද you have intergenic. Right? Intergenic. Genes දෙකක් මැද intergenic. Okay? You have something called intergenic DNA. ඒක කොළ පාටින් කියන්න. Intergenic DNA. Then within a gene, when you consider inside a gene, right? When you consider inside a gene, so make up a logo karo. And you take the inside of the gene. When you take the inside of the gene, then molecular biology but usually a gene is transcribed, right? A gene is transcribed. Transcribe karna කියන්නේ මොකද්ද? DNA එක mRNA එකට convert කරනවා. That's transcription නේ. That's transcription නේ. Yeah, that's transcription නේ. So a gene is transcribed, but not all parts are translated. Translated කියන්නේ මොකද්ද? Converting the mRNA into a protein. So this is the mRNA molecule. Okay, this is the mRNA molecule, and within the mRNA molecule you have two more regions, right? Two more regions. The regions which are translated into proteins are called what? The regions which are translated into proteins are called what? The regions which are translated into proteins are called what? හිතාගන්න බෑ අරකද මේකද කියලා. අරකද මේකද කියලා හිතාගන්න බෑ. යයිද මේ දෙකෙන් එකක් නේ. මොකද්ද? What are translated? Yes. ඒක මද තියන විදිය තමයි දැන් මෙතන ජීන් එක මැද තියෙන ඉන්ටර්ජෙනික් DNA ඉන්ටර්ජෙනික් I එක ඉන්ටර්ජෙනික් වෙලා I එක ෆන්ක්ෂන් එක නෑනේ අපි දන්න තරමේ ෆන්ක්ෂන් එක නෑ ඒ වගේ in the gene the introns the one with the I doesn't have a function එහෙනම් translate වෙන්නේ කවුද translate වෙන්නේ exons right exons තමයි translate වෙන්නේ ඔය translate වෙන්නේ නැති ටිකට අපි කියනවා introns කියලා මතක තියාගන්න I මුලින් එනවා නම් හරියම function එකක් නැහැ. දැන් මෙතන උනත් even though it is transcribed, it's not translated. So again, no promising function නේ. So අනේ මතකයි. Okay. Anyway, when we consider this intergenic DNA, මම අර යට කැලේ නම් කියලා දුන්නේ to just test your knowledge on these molecular biology, and not very good it seems. Right? But අවුලන්නේ shape. At least you know the terms නේ. At least you know the terms. That's more than enough for now. Right? At least you know the terms. Right. So when you consider this intergenic DNA, at first it didn't have a known function, right? Intergenic DNA වලට known function නැහැ. Basically ඔය intergenic DNA වල ඉන්නේ මේ small tandem repeats හදන්නේ STRs හදන්නේ intergenic DNA වල, right? So it didn't have a common function or known function. But this human genome sequencing has like identified some function where it might be it acts as these regulators. They regulate the genes, right? They regulate the genes and the transcription process and all that. They involve in regulation. But ඊට වඩා තාම function එක හොයාගෙන නැහැ. But still it is being found. Again, the human genome project is still being going on where the function of the intergenic DNA is still being found. මොකද large portion of our DNA is intergenic DNA. Okay. So බලන්න. The genome projects will have additional benefits that at present can only be guessed at. It is evident that the human genome, in common with the genomes of many other organisms, contain extensive amounts of intergenic DNA. It was thought that most of the intergenic DNA has no function except occasionally it is known that some intergenic DNA acts to control genes nearby. So regulatory function at Tibba. Could the intergenic DNA have a role? One that at present is too subtle for us to grasp. So there might be a function where it is not found yet. Though little is known about them, they are thought to have regulatory functions. The first step in addressing this possibility is to obtain a complete description of the organization of the intergenic DNA in different genomes, so that the common features which might indicate a role for some or all of these sequences can be identified. So the common features, if they are identified, sometimes a role can be identified. Right. So the lesson is completed. Was it clear? Human genome project එකත් අවුලක් නැද්ද මම දන්නා හෙන හැලි ගොඩක් තියෙනවා ඒව ගැන ලොකුවට හිතන්න එපා මම point out කරපු points ටික විතරක් පාඩම් කරගන්න. Was it clear? In the explanation, was it clear? What about the others? Was it clear or not? Any questions? Right. Yes, sir. Patta clear. Thank you. Thank you, sir. Okay. Thank you for the comment. Right. Let's do some questions and see what type of questions can be asked. So this came in the 2023 A-level exam. Try this question. Is this human genome project still undergoing? The human genome project was completed, but still it's being new stuff is added into the project. And it was completed in 2003. Still some new updates are being added as well. Molecular biology, I did one seminar but I did the molecular biology lesson for the 25 batch. So ප්‍රශ්න දැක්කම ඒ level අපේ අවුරුද්ද ප්‍රශ්න මත් එකපාරට බය වුණා. Even I didn't study the human genome project that much, but basically using the practical knowledge ඔක ගන්න පුළුවන්. Right, let's see. So May question for pay avurudinaave. So when I saw this question, the only two things that I remembered was I remembered E. coli genome project was already completed and the human genome project is already completed. මම දැන් හිටියේ B and D විතරයි. So ඒකේ ඇත්තට උත්තරේ ගන්න පුළුවන්. මේ so දැන් ඔය මම මේ ටික පාඩම් කරන් ගියා. E. coli yeast human genome project එක. So තියෙනවනේ මෙතන the human genome project and other genome projects such as E. coli yeast, mouse, Arabidopsis thaliana, rice have already been completed කියලා. එහෙනම් ඒකෙන් කියන්නේ මොකද්ද? Genome project for E. coli is already completed. D is correct. Human genome project is yet to be completed. වැරදි. So එහෙනම් B is wrong, D is correct. එහෙනම් from that first answer is gone, second answer is gone, third answer is gone, fourth answer is gone. So ඉතින් පහ ලාලා ආවා. Right now that I remembered all the four. But I think if you know that B is wrong, you can answer all you can directly get the fifth answer. Right? So anyway, A බලමු අපි දැන් ඉගෙන ගත්තනේ. So A, one of the aims of the Human Genome Project is to determine the sequence of 20,000 base pairs. No, it's the 3 billion base pairs. 3 billion base pairs and 20,000 protein coding genes, right? 20,000 protein coding genes and 3 billion base pairs. Human Genome Project has led to the description of molecular activities of human cells. Yes, right? Advancement of molecular medicine or something. So anyway, molecular activities can the DNA sequence is a molecule which is present in our cells. So it gives a description of the molecular activities of the human cells. So it's C and D, fifth one. Right, let's see some more questions. Try this question and send me your answers. Let's see. Identify the incorrect statement with regard to the Human Genome Project. And aim of the project was to identify all the genes in the human genome. Yes, that was the name of the project. According to a major quality assessment, only 92% of the sampling exceeded 99.99% accuracy. No, that's wrong. Over 99%. I think over 99% of sampling exceeded 99.99%. See, small words matter a lot, right? So according to a major quality assessment, only 92% of the sampling exceeded 99.99%. So that's wrong. That's incorrect. Not only over 92%. Okay. Genome projects of E. coli, yeast, mouse, Arabidopsis thaliana, and rice have already been completed. Yes. Intelligent DNA is thought to have regulatory functions. Yes, it is thought to have regulatory functions. Though little is known about them, they are thought to have regulatory functions. Study of human evolution and anthropology is the benefit of sequencing of the human genome. Yes. Right. So the second one is the answer. Right. Second one is the answer. Right, try this question as well. Right. The Human Genome Project pursued a wide range of ambitious goals according to the note. Which of the following statements incorrectly describes one of the Human Genome Project's primary aims? To determine the precise sequence of all 3 billion chemical base pairs that constitute human DNA. Not for the chemical base pair. Determine the sequence of 3 billion chemical base pairs that make up the human DNA. Incorrectly. Yeah, incorrect. That's so 3 billion base pairs that constitute human DNA. That's correct. To identify all the genes within the human genome, excluding the protein coding genes, including the protein coding genes. Yes, right? So, identify all the genes. To ensure all related genetic technologies are exclusively owned and controlled by the public sector, preventing private use. No, to provide the private sector these technologies as well, right? These technologies as well, because the private sector is what's more developed, right? So they need to provide the private sector with these technologies as well. So that's true. To establish and improve efficient databases, yes, to actively address and explore ethical, legal, and social issues that could arise from the project's findings, yes. So the third one is the best answer. Right. Let's quickly watch a video on the Human Genome Project.
An organism's genome is the entire genetic material of that organism, so the complete set of DNA. It provides all of the information required by the organism to function. DNA is a long chemical that contains bases represented by the letters A, T, G, and C. A gene is a section of DNA that codes for a particular protein. To learn more about DNA, watch this video. The human genome is 3 billion bases long, but only consists of about 20 to 25,000 genes. As an average sized gene is only about 3,000 bases long. This means that large parts of our DNA doesn't code for genes. The genome of every person.
So that now in the human genome there are 3 billion genes. So they divide most of the DNA in the human body is intergenic DNA, right? It's intergenic DNA, not genes. So each gene is made up of 3,000 base pairs, and there are only about 20,000 genes, right? So anyway, you can see that most of the DNA in our body is intergenic DNA.
Person on Earth is 99.9% the same. It's that tiny 0.1% that makes up genes that give us our unique differences. In fact, our genes are 98% similar to a chimpanzee's and 75% the same as a mouse. The Human Genome Project was an enormous project that saw scientists around the world collaborate to work out the sequences of bases in the human genome. The project started in 1990 and was completed by 2003, 2 years ahead of schedule. And here is the order of the bases: A, T, G, C, T. Only joking. If I was to read out the bases, one at a time, for 24 hours straight, it would take a whole century, and I certainly don't have time for that. So, now we have the information, how is it being used? Well, since the structure of DNA was determined in 1953, we have known that the order of the bases is very important, and that a change or mutation in this order can result in diseases. By sequencing the whole genome, scientists can use this information to work out the genetic causes of diseases, such as cancer, as well as developing treatments.
So they can basically, again, they can determine these DNA sequences of these diseases, cancer cells, genetic disorders, by this Human Genome Project.
The forms of genes, or alleles, associated with different inherited disorders have been mapped, which means that genetic tests can be carried out on people to see if they are sufferers or carriers of an inherited disorder. For example, a genetic test can tell if someone is a carrier of cystic fibrosis. By comparing the DNA of people living in different parts of the world, scientists have been able to trace human migrations. The Out of Africa theory, that the first humans left Africa within the last 100,000 years, has been supported by comparing the DNA of people living in different areas of the globe. The evidence shows which people are more closely related to others. Because of all the data generated by the Human Genome Project is available to anyone to view free of charge online, means that scientists around the world can use the information in their research. This helps to increase the number of uses for it. There have been major advancements in DNA sequencing since the Human Genome Project ended. It took 13 years and billions of dollars to sequence a whole genome during the project, but now scientists can do it in a matter of hours, and you can even get yourself sequenced for a small fee. These advancements will help develop new strategies for the prevention and treatment of disease. So, in this video, you've learned what a human genome is, what the Human Genome Project was, and how the information from it
If you like the video, give it a
Right, so it gives a description on how the Human Genome Project was helpful, and also finally here you can basically sequence your own genome within a matter of hours. I'm not sure it's in Sri Lanka, but in foreign countries you can usually do that, right? So one more video before we go, which I actually forgot to show you guys, about nanotechnology, right? ඒකත් බලලා මම ඉතින් අපරාධනේ මෙච්චර කරපු එකේ.
How small is the smallest piece of technology you can imagine? The size of a grain of rice? A grain of sand? How about the width of an individual strand of spider's web? A red blood cell? Narrower than a single wavelength of visible light? Okay, now we're talking. Welcome to the realm of nanotechnology. At this minuscule scale, we measure things in nanometers. To give you a rough idea of just how small those are, in one day, your fingernails will grow by around 86,000 nanometers, about the width of a human hair. By comparison, nanotechnology is defined as being between 1 and 100 nanometers across, which is just a touch more than your fingernails would have grown since you started watching this video. Despite the term nanotechnology having been in common use since the 1980s, with some of the underlying ideas first being proposed in the 1950s, we've only just started exploring the benefits of utilizing such a small scale. Even though it carries a sense of futuristic excitement, it's worth noting that it's already all around us. It's just important to realize that by technology, we don't mean anything with a circuit board, but rather the far broader definition of the application of science.
Too fast, ne?
Scientific knowledge for practical purposes. Let me give you an example. When you next head out into the sunshine, take a second to think about the nanoparticles in your sunscreen, reflecting harmful UV rays, or those which make up the scratch-resistant coating on your sunglasses. From microphones to aerogel insulators to tennis rackets to LCD monitors to cancer treatments, nanotechnology has been part of our everyday lives for longer than you think. In actual fact, it's thought that the legendarily strong steel blades produced in Damascus around the 12th century owed their strength to nanotechnology. Unknown to the Syrian blacksmiths, the repeated heating and cooling of the steel during forging caused impurities in the metal to separate, resulting in the creation of carbon nanotubes.
So this is how carbon nanotubes were created. Basically, when these metals were purified, these carbon nanotubes were created, and carbon nanotubes are actually much stronger than steel and very low in weight. right? So you'll see it.
which today are one of the fundamental building blocks of nanotechnology. Carbon nanotubes are, unsurprisingly, tiny tubes made of rolled up sheets of carbon a few nanometers across with a honeycomb-like structure. They can be created in a bunch of cool, sciencey ways by inciting reactions in graphite using gas, by blasting it with lasers or plasma, or zapping it with electricity. Tricky work, but still much easier than rolling them by hand like some kind of microscopic sushi chef. The really cool thing about carbon nanotubes, though, is that if you roll them up just right so that the carbon atoms align in a certain way, you have a tube that is 100 times stronger than steel, but 6 times lighter. And the potential construction applications of a material like that are mind-boggling. What's more, if you roll them in a slightly different way, carbon nanotubes can conduct electricity, which means that by coupling them with nanowires, scientists are well on their way to making tiny computer chips. Tiny computer chips equals tiny computers equals tiny robots. And all of this starts to paint a picture of a future full of scientific possibilities. Consider the idea of injecting blood cell-sized nanobots into a patient's bloodstream to fight infections and repair brain damage, or creating nanofabricators, 3D printer-like devices building on an atom-by-atom basis to produce anything from a...
So there's a 3D printer is basically something which is not developed yet. 3D printer can basically using these nanoparticles. They are in the molecular level. You can print items, that you can just print items. That's at a 3D print is but using nanoparticles, you can start from the molecular level and build these 3D items.
A chunk of coal to a glass of wine. And that's before we talk about invisibility cloaks or space elevators, both of which are real projects utilizing nanotechnology and being worked on as we speak. Now, inevitably, for such an exciting, experimental, and potentially disruptive technology, nanotechnology comes with some areas of concern. Nanoparticles are classed as UFPs, ultra-fine particles. We've only just begun research into how to regulate these as nanotechnology evolves. There are medical concerns. Could nanoparticles cross into your bloodstream or into your brain? There are security ramifications. The idea of nano-surveillance devices is fairly terrifying, and alongside those are moral quandaries. If a nanofabricator gives you the ability to build anything, what stops you from building a lethal weapon? How could technology like that affect the manufacturing industry or the global economy? These are all reasonable questions that right now
It's used for cooking 3D printing food. 3D printing food. It's designed cooking, I don't know. Like you can print food, as in you can print a cake or an apple, something like that. Okay. For decorations. Okay. I think I know what you mean. Edible deco. Okay. Plastic printing. Okay. Right. Here, Lanka, I didn't know that. So, yeah, now the lesson is done and we discussed many questions as well. And Mama may structure design may discuss because you can just find answers for the structure essays directly from the resource book. But I will upload a document with the MCQs again. The MCQs and the structure essays. I'll upload a document with that so that you can do them yourself. The classified structure essays from the past papers. So now in 2023 and 2024, let me quickly show it to you. I didn't make a good one. Classify I'll quickly show it to you. 3D printing of cigarettes. Not this one. So, wait. Right. So 2021, you just got 2 questions. Concept, main concept on which tissue culture is based, and how does addition of sugar preserve for? Then monkey 2022, you got 1/3 of a structure essay. So complete part C is from applied biology, right? So 4 issues faced by growers, or issues by gelling agent used during tissue culture medium. What's gelling agent use? I mean, body questions. Gelling agent added for tissue culture medium. What is it? Yes, that is it. I think we did this about 2 weeks ago. Okay, good. It's AGA, right? AGA. What are the conditions that a seed has to complete to initiate germination? So, three principles of food preservation. We even had an MCQ from that. Role of soil reproduction of dry fish, known as nanoshells, state two uses of nanoshells. So, role of known as nanoshells. These nanoshells are these nanoparticles which have a dielectric core, and two uses treating cancer and for bioimaging enhancement. And 5 benefits of sequencing of human genome, right? 5 benefits. So AK 2022 level. Let me quickly show the marking scheme. They are not asking about the aims, okay? Not asking about the aims, they are asking about benefits. So අපි මම වලට පෙන්වන්නේ වෙනම benefits කියලා කැලලක් පෙන්වන්නේ. Right? So in the marking scheme, what they have given is and then improved diagnosis of various diseases, identification of mutations linked to cancer, design of medication, gene therapy, study of human evolution anthropology, control system of drugs, right? So know these in may benefits ටික මතක තියාගන්න බලන්න. ආය අහන එකක් නැති වෙයි ඒත් කියන්න බෑ, right? කියන්න බෑ because past paper questions are repeated, right? Past paper questions are repeated a lot. So this question might come to your IELTS as well. මොකද මේක ආවේ 22 වලනේ. So 23, 24, 25. Okay. Then 23 wala state how BT toxin affects mosquito larvae. That's easy. Basically, BT toxin will lyse the tissues of mosquito larvae. But again, what organism produces the BT toxin? Who's the organism which produces the BT toxin? The organism which produces the BT toxin? Yes, right. It's the Bacillus thuringiensis islanensis. Can't full name, but Bacillus thuringiensis keyword. Right? So it's the Bacillus thuringiensis. Israelensis is the full species name. So BT toxin yeah. And 24 levels again you got just one small question. Two uses of nano devices. So two uses of nano device sensors. Yeah. So nano device sensors. Okay, answer answer it quickly. One by answer, state two uses of nano device sensors in nano medicine. Other correct. Nano device sensors are uses tuna. Tunak tibba. Dekak kiyan. Nano device sensors. Yes, tunak tibba. Dekak kiyan ikante. You can find it here. Nano device sensors are used to monitor blood pressure, blood oxygen levels, and hormone concentration. So, ඔය තුනෙන් ඕනි දෙකක්. Right? ඔය තුනෙන් ඕනිම දෙකක්. Diabetes වලට අදාළ නැහැ. Okay? Nano device sensor එක ගැන ඇහුවොත් you need to mention only these three. Blood glucose level ලියන්න එපා. It's not given in the resource book නේ. Right? It's not given in the resource book නේ. දීලා තියෙන්නේ මොනවද? Is used to monitor or measure or detect blood pressure, blood oxygen level, and hormone concentration. So ඔය තුනෙන් එකක් විතරක් කියන්නේ blood glucose level නම් ලියන්න එපා, ලකුණු හම්බෙන්නේ නැහැ. Right? So blood pressure, blood oxygen level, and hormone concentrations. Right? So structured ආවේ එච්චරයි. But 25 වල ඔයිට වඩා ටික expect කරනවා. Because with the papers becoming more difficult and difficult, they might add more structure, right? From unit 2. නෑනේ ළමයා මේ තියෙන්නේ diabetes nano devices. Okay, the word is nano devices. In the question, what was it? The question was nano device sensors. Okay, the question was state to uses of nano device sensors in nanomedicine. Okay, so nano device sensors nano devices you can write glucose. Okay, nano devices you can write glucose as well, the insulin secreting thing. But nano device sensors write only that. Right. Okay, so with this lesson, I have completed the syllabus for the 25 batch, right? So the syllabus is complete for the 25 batch. So was this lesson clear? Was this lesson clear? බඩගිනි නේද? මඩක් බඩගිනි. උගලන්ට කන්නයි උගල මට කන්නත් බෑනේ උගලන්නේ. Right. Okay. Okay then. Okay. Some good comments as well. Right. Okay. So I hope the lesson was clear because this lesson is not done much by teachers. So I took my time and completed the lesson and explained the parts which I hope is clear. And we did so many questions as well, right? We did about 50, more than 50 questions from applied biology. MCQs godak tara, right? So just generally read through the lesson and try to study the important points that I pointed out before going to the lesson, because usually at least minimum MCQ degakwa tahana, and they are not that difficult as well. We did the past papers, right? So just definitely score them, right? Definitely score them. Right, okay. The recordings are also available in the website and I'll be uploading it to YouTube as soon as possible as well. So, okay then, I'll end the meeting then. Good night, everyone, and thank you for joining.